Dr. Nelson Moussazadeh, who's recently joined MD Anderson from Sloan Kettering, where the Chordoma Foundation has had a long-term, very productive collaboration. I've been very pleased to support Dr. Moussazadeh's work. He's going to talk to you about findings with respect to epigenetic analysis of chordoma. So not the actual DNA sequence itself, but the configuration of the DNA and different marks and modifications that are on the DNA that occur in the tumor. So, Nelson, it's all you.
Well, thank you very much, Josh, for the kind introduction and for putting on another spectacular Chordoma Community Conference. I always very much look forward to these meetings, in particular because the patient population and the caregivers here are just so incredibly sophisticated and engaged in their disease, and it's really a tremendous inspiration to our work, both clinically and on the research side. So this, I apologize in advance, it's a technical molecular biology talk, so please be patient. It's a talk about epigenetics. So just as a brief reminder from high school biology, the genome is the DNA code that we each have in each of our cells. It's the blueprint that makes us who we are. It's very, very long, 3 billion letters long. And the Wellcome Trust actually had it all printed out in book form, and it was about 262,000 pages long. And despite each of our cells in our body having an identical genomic code, even within the same person, all of the cells behave very differently. So we have brain neurons, we have heart muscle cells, we have skin cells, and these cells behave extremely differently despite having exactly the same genetic code in every cell. And the reason for this is the epigenome, or super important switches in the DNA that actually turn different proteins on and off. And it's the proteins themselves that actually govern how cells act. This is a very complicated field, and I'm going to focus on some of the most important epigenetic switches, which are called histones. And histones are a class of proteins that dictate which pages of the genome book are read and acted out by a given cell. So this particular cartoon shows how histone proteins, which look something like macaroons that entangle themselves into chapters of the DNA code. So here you have the string, which is really just this incredibly long length of genomic code that's entangled in these proteins. And they essentially make areas of the DNA open or closed, and that is available or unavailable for reading into proteins, which then go on and have the, enable the cell to act in different ways. And the histones themselves are complicated. The way that they interact with the DNA is governed by, they use what we call bookmarks, so special decorations on the proteins themselves that allow the histones to interact with the DNA in different ways. And different cells have different chapters open at any given time. And this is due to the histones themselves having different bookmarks on them, which allow the DNA to be open or closed in particular times and in particular places within their genome. There's an entire field dedicated to understanding how histone bookmarks behave and how and when they're placed in given cells. This cartoon is very complicated, but it shows a list of genes. They're listed in red and green. That themselves place and remove different types of bookmarks on the histones, telling each cell where and when to read different parts of the genome code. And as you can see, it's a very complicated process. There's a lot of different genes involved. There's a lot of different histones and different places in the histones that can be affected. And as with any complicated process in cells, things can go wrong. And very commonly, when things go wrong in histones, this can contribute to cancer. So this cartoon is a recent figure showing how many different, or in how many different cancer types, mutations in histone proteins actually contribute to cancer. So here you have a list of cancers ranging all the way from leukemias down to renal cell carcinoma, breast cancer. And this is very exciting because this may lead to new targets for therapy. So the question becomes, are there errors in these histone bookmarking processes in chordoma? There's a few reasons to suspect this. First of all, as we heard before, chordoma behaves very much like a specific mature cell type. So they, outside of the chondroid and dedifferentiated types, chordomas look very, very similar pathologically from patient to patient. And they look very similar also to an embryonic cell type, the notochordal cells. And cell type is governed by epigenetic state. So that's one reason to suspect epigenetics here. Another reason is that drugs have actually been tried that target histones. And so histone deacetylases have been used in cell lines, and they actually cause histone cells to senesce in vitro. And the third reason is that chordomas have a gene, Bracari, which seems to be stuck in the on position. So we've heard already that there's duplications, there's focal amplification sometimes, and there's a SNP, or there's a polymorphism that's seen in every one of you patients in this room, most likely, that contributes, but it's not responsible. We don't have a mutation that turns Bracari on in a very strong way that causes chordoma by itself. Out of those of us who don't have chordoma, half of us have this SNP, and it's not enough. So is there an epigenetic mechanism causing Bracari to be in the on position? We'll hear more from Dr. Lin, who's much more of an expert in that domain, but it definitely suggests that there's an epigenetic role here and a potential vulnerability in chordoma. And in fact, we already heard from Dr. Wang that chordoma is extremely lightly mutated. So this figure just shows, on the bottom line, we have a list of a whole range of cancer types. And on the y-axis, we have frequency of mutations, and it's actually a log scale, so every notch is 10 times more mutations. And you can see chordoma is at the very bottom of the spectrum. There's only about 15 mutations when you sequence someone's exomes or set of the exon part of their genome. So 15 mutations is really very, very little, which suggests that it's not just mutations. There may be an epigenetic phenotype, an epigenetic phenomenon going on here as well. So our research has been focused on identifying whether there's a specific histone pattern in chordomas, a bookmarking pattern, and if so, why? And most importantly, how can we translate this into drugs that can treat all of you and all future patients with chordoma? So to start to look at this, we did a pilot project at my previous institution, Sloan Kettering. We looked at genes that dictate histone bookmarking. So the one I showed you this figure before, the red and the green are all the genes that we know of that can affect this bookmarking process. And we found mutations, interestingly, in many of these. Not many of them were in the same genes, but it was interesting to see that many different histone processes were being affected in different patients. We saw this figure before also, but in the most definitive genetic study of chordomas that came out of the UK just a couple months ago, this finding of mutations in histone-modifying genes was real, and it included, there's a couple genes listed here, but one of them, interestingly, is SETD2. I'll come back to this in a minute. Our collaborators at Sloan Kettering looked at gene alterations in a different way. So I showed you mutations in the last figure, but another way of looking at gene loss is to look at copy numbers. So this study done at Sloan Kettering looked at copy numbers, so it looked at deletions, and it was reported that about 2/3 of patients had loss of a region of the genome that has multiple epigenetic genes, including SETD2 and polybromo1, which are both found to be lost by a different mechanism, by mutation, in chordomas. So this led us to ask the question, what does the histone bookmarking pattern look like in chordoma? Is there a specific pattern across all chordomas in their histone phenotype? And if so, can we identify why? So to do this, the old way of looking at histones is cumbersome. So it uses antibodies, you look at individual bookmarks, but there's hundreds of them. And as you all know, chordoma tissue is rare. Most patients hopefully only have one surgery, but there's not many of you. So getting tissue is very valuable, and we want to be as efficient as possible in looking at these tissues. So in the first time that anyone has used mass spectrometry, where you look at many hundreds, potentially, of histone bookmarks, we were able to quantify the specific bookmarking pattern across different patients' chordoma genome. So this is a complicated figure, and I'll try explaining in a minute. But what we found is that the bookmarking patterns in chordoma are remarkably similar from patient to patient. So this was very interesting. It suggests that histones may actually play a very important role in maintaining a very specific pattern of cell behavior in patients. And they're also very different from other cancers. So we all know that chordoma behaves differently from more malignant cancers. It's very indolent. It tends to grow in one set of places. It's locally aggressive, but it tends to not metastasize. And you can see here, so I'm going to try and explain this figure, so bear with me. It shows, basically every box that you see shows a different type of bookmark. And the pink boxes are chordomas, and the green boxes are other cancer types. And when you compare them, this is one particular bookmark that's very interesting because there are several other cancers that have the bookmark all the way on the right in a low level. In chordoma, they're not in a low level, which actually suggests that there's not too much reading activity of the genome due to this particular bookmark. This is another set of bookmarks. This was a little more interesting for us because they lead to high reading levels in certain parts of the genome code. And it was particularly interesting because this particular bookmark would be explained by the deletions in SETD2, which I showed you before, which occur by both mutation and by deletion. And this is a different way of looking at bookmarks. So this is using an antibody, looking at different cells, under the microscope. And you can see in the middle panel, this is a chordoma looking for this particular bookmark. Turns out it's gone in patients that have loss of SETD2. So this was very interesting. And finally, I'm just going to show you one last type of bookmark, which we know is turned on by brachyury, this all-important transcriptional factor. And indeed, we find that it's turned on across the genome code in chordoma cells. And this is very different from other cancer types. So all of this work is important because we know that drug companies are now working feverishly to identify drugs that place or remove these different histone bookmarks. And while we all know they're driven by profit and they're probably not looking specifically for chordoma, we also know that if we understand how these work in chordoma, we'll be primed to apply the relevant drugs as soon as they become available, if this indeed is an important mechanism in chordoma. I'll just finish by saying that, importantly, we're not just waiting on drug companies. We're also looking, with the help of the Chordoma Foundation, at identifying vulnerabilities ourselves. And this is an experiment that we're doing, looking at over 500 genes that affect epigenetics. All the bookmark mediating genes that I showed you in earlier slides, as well as another 2,000 genes that we know have available drugs. And we're doing a very high efficiency experiment where we're looking in cell lines at thousands of these different genes, turning them off at the same time, and having the cells compete against each other in a single experiment with one large pool of cells and see which cells actually do the worst with individual genes turned off. This is a very exciting type of experiment because for the first time we can actually look at many thousands of genes all at the same time with built-in controls in a robust way. And we're hoping that it'll help us identify needle in a haystack type genes that will be a very high value for targeting in future studies. And our approach is very exciting for me because we're finally able to leverage different cell lines. So cell lines, as you know, are of super high value in chordoma, and thanks to the foresight of Josh and the foundation, there's been a tremendous expansion of cell lines that we can now use in our in vitro experiments that can inform future patient-driven studies. And we're actually able to leverage differences in different cell lines to find the highest value targets. So in this particular figure, you can see individual dots represent different genes that we're looking at. If we shut them off, what happens to the cell growth? And in this particular figure, we're looking at two different cell lines. The cells on the green dots on the left represent genes that very much hurt one cell line, and dots on the bottom are genes that hurt the growth of a different cell line. And now that we have two different cell lines that we're able to grow very quickly in the lab, we're actually able to prioritize genes that affect multiple different cell lines. And of course, now I heard from Josh last night that we're up to, what do we say, 20 different cell lines now finally that we can use. We are aggressively looking to validate our highest priority targets in all of the cell lines, but in a very focused way. So we can pick our 10 best genes in the epigenetic and drug available domains for prioritization. And we hope to work and partner with the foundation and getting these into animals and patients just as soon as we find something that's promising. So thanks so much to the foundation. Thanks to my collaborators back in New York and Chicago and here. And thanks to you all for your attention.